Primeval, The Dark Continent, by Bryson Roberts, read by Roger Cartwell. Everything is theoretically impossible until it is done. Robert A. Heinlein, 1952. Cloning will enable mankind to reach eternal life. Claude Vorillon, 2001. Genetic engineering has never been about saving the world. It's about controlling the world. Vandana Shiva, 2005. In pushing other species to extinction, humanity is busy soaring off the limb on which it perches. Elizabeth Colbert, 2014. Introduction. Innovation is key. The science of today becomes the technology of tomorrow. That was what Edward Teller once said, and if history is any guide, he was right. The 21st century has brought with it an avalanche of technological advancements. The internet has become the beating heart of innovation, an endless frontier where ideas can grow and thrive. The smartphone has tethered us to the world, shrinking distances that once separated us. What once seemed impossible, instant communication across the globe, is now an afterthought, embedded into daily life. But with these advancements, a new, more unsettling frontier has emerged: genetic engineering. While it promises to reshape life itself, we are only beginning to grasp its potential consequences. The world is changing, and nothing will ever be the same again. Our story begins with Gregor Mendel. In 1865, Mendel's experiments with pea plants laid the foundation for genetics, though his work went largely ignored during his lifetime. It wasn't until decades later that his theories of hereditary segregation and independent assortment would be recognized as groundbreaking. By the early 20th century, Mendel's work had sparked a revolution, and biotechnology was born. Laboratories around the world raced to unlock the secrets of life, transforming what was once science fiction into scientific fact. In 1972, Paul Berg achieved a monumental breakthrough, combining DNA from a monkey virus with that of a bacterial virus. Berg created the first recombinant DNA molecule, a feat that would soon be surpassed by Herbert Boyer and Stanley Cohen. Using a restriction enzyme to cut a bacterial plasmid and insert foreign DNA, Boyer and Cohen produced the first genetically modified organism. Their creation, bacteria capable of surviving in the presence of antibiotics, marked the dawn of a new era, one where life could be engineered, redesigned at will. Not long after, in 1974, Rudolf Jenisch produced the first transgenic animal, a mouse with foreign DNA integrated into its genome. Jenisch's discovery paved the way for genetic manipulation in higher organisms. By 1981, scientists had demonstrated that these transgenes could be passed on to subsequent generations, transforming theoretical genetics into a powerful tool for altering life itself. But as genetic technology advanced, so too did the ethical and moral questions surrounding it. By the early 2000s, genetic engineering had shifted from bacterial cells to plants, and even to human embryonic cells. In 2010, scientists at the J. Craig Venter Institute created Synthia, the first synthetic life form, pieced together from artificial DNA and brought to life in a laboratory. The implications were staggering. Life, once considered a force beyond human control, had become a system we could manipulate, modify, and even create from scratch. Then came CRISPR. By 2015, this gene-editing technology had ignited a firestorm of debate. Scientists could now edit plant and animal genomes with surgical precision, splicing out unwanted genes or adding new ones to improve crop yields, make animals disease-resistant, or perhaps more controversially, modify human embryos. CRISPR offered the promise of a future free from inherited diseases, but it also opened the door to genetic manipulation on a scale never before imagined. Could we engineer our children to be stronger, smarter, more resilient? Could we alter the very fabric of humanity? The backlash was swift. Environmentalists warned of the dangers posed by genetically modified organisms, GMOs, while bioethicists raised concerns about the moral implications of genetic engineering. Could these technologies, they asked, create unforeseen risks to human health? Could they disrupt ecosystems, leading to unintended consequences? Reports surfaced of animals fed GM crops suffering from infertility, early death, and mysterious health problems. While many studies disputed these claims, the controversy deepened. The CRISPR technology that had promised so much was reshaping agriculture, medicine, and science. Chinese researchers were using it to boost rice yields and create fungus-resistant wheat, while British scientists were tweaking barley to better withstand drought. It was hailed as a breakthrough, but at what cost? The U.S. Department of Agriculture began exempting certain gene-edited crops from regulations, and the line between nature and technology blurred further. At the heart of this genetic revolution lay one uncomfortable question: who decides what's right and what's wrong? Who determines how far we should go in reshaping life to suit our needs? The controversy reached a fever pitch when scientists began editing human embryos. What if parents could choose their child's eye color, height, intelligence? What if we could build our children the way we build our homes, picking traits from a genetic catalog? It was a terrifying thought, and one that no longer seemed distant. As scientists pushed the boundaries of genetic modification, they also revisited a long-forbidden dream: cloning. In 1996, the world met Dolly the sheep, the first mammal cloned from an adult cell. Dolly's birth sparked debates that continue to this day. Could we one day clone humans? Should we? Cloning technology advanced rapidly, and by 2013, researchers had successfully cloned human embryonic stem cells. It was a breakthrough that held immense promise for medicine, but it also revived fears of a future where human beings themselves could be cloned, altered, or even mass-produced. But all of this paled in comparison to what was happening on a remote speck of land in the Andaman Sea, deep in Thailand's Trang province. The island did not appear on any navigational charts. To the fishermen who occasionally glimpsed its jagged silhouette, it was simply Ko Sangraam, the island of conflict. The name was a warning passed down through generations, muttered over nets and diesel engines in hushed tones. Old men swore the waters around it were cursed, a place where compasses spun erratically and strange shadows moved beneath the surface. To the Thai government, it was something far more benign, a restricted maritime zone, designated on official maps as an environmental research reserve. The wording was vague by design, an elegant fiction buried in bureaucratic language. The truth was infinitely more dangerous. Ko Sangraam was ground zero for the most audacious biological experiment in human history. Genova Corporation was not a household name. It was never meant to be. Publicly, the company operated under a thin veneer of respectability, an innovator in life sciences, they called themselves. In press releases, Genova spoke of breakthroughs in cancer therapies, advances in crop resilience, and pioneering conservation programs designed to save endangered species. They funded conferences, they published white papers in glossy journals, they cultivated an image of quiet benevolence. But beneath the surface, Genova was something else entirely. Founded in 1986 by Swiss-born industrialist Victor Kessler, Genova had begun as a modest agricultural genetics outfit operating out of a Zurich laboratory the size of a small apartment. By the 1990s, the company had grown into a sprawling multinational with private facilities on 3 continents, and with that growth came a change in vision. Kessler's interests shifted from seeds and soil to something far more ambitious: the resurrection of extinct life. It began, as these things often do, with data. The late 1990s marked a turning point in molecular biology. Polymerase chain reaction, PCR, had already revolutionized the ability to replicate fragments of DNA. But it was nanopore sequencing, introduced in the final years of the millennium, that changed everything. Suddenly it became possible to thread ancient genetic fragments, tiny, fragile strands of nucleotides, through nanoscale pores and read them base by base. At first the results were modest: scraps of genetic material from frozen mammoths, shards of sequence from desiccated cave bears, partial genomic reconstructions of Pleistocene horses. The work was slow, excruciating. Contamination was constant, degradation unavoidable. The scientific consensus was firm: de-extinction was a fantasy. But technology evolves faster than consensus. By 2010, the introduction of single-molecule real-time sequencing shattered the old barriers. Now, entire genomes could be assembled in hours rather than years, using strands so degraded they were once thought worthless. Suddenly, paleogenomics wasn't theoretical. It was actionable. The implications were staggering. In the hands of academic teams, the advances were couched in cautious language: population genomics, phylogenetic inference, conservation genetics. But inside the boardrooms of Genova Corporation, the language was different. Their internal white papers, classified at the highest security tier, were blunt. Complete genomic reconstruction achievable within 5 years, full de-extinction within 10. The first steps were small, almost benign. They targeted Holocene extinctions, species that had vanished only centuries ago: rodents, lagomorphs, small ungulates. The press releases were upbeat, draped in the language of ecological ethics, rewilding initiatives, biodiversity restoration, environmental stewardship. No one asked too many questions. The world was desperate. Forests were vanishing, species were collapsing, and climate change was rewriting every ecological script on the planet. If Genova claimed to have solutions, people listened, and they paid. But ambition is a moving target. By 2013, Genova had created an entirely new division, the Paleobiological Systems Division, under the leadership of Dr. Franz Reinhardt, a German-born geneticist with a reputation for brilliance and an equal reputation for ignoring ethical boundaries. Reinhardt was more than a scientist. He was a zealot, a man who believed extinction was a solvable error. Reinhardt pioneered what he called predictive genomic assembly, a process that fused CRISPR-Cas gene editing systems with probabilistic modeling and synthetic scaffolding. Where ancient DNA was fragmented, as it always was, his algorithms didn't merely patch the gaps. They rebuilt them using evolutionary prediction models, drawing on reference genomes from living relatives, and then improving them. This was no longer restoration. It was enhancement. In an internal memo dated February 2014, circulated only to senior executives and marked eyes only, Reinhardt wrote a single chilling line: "Extinction is an error state Evolution is an unfinished program. We have the tools to debug it. Those words became doctrine inside Genova's inner circle. Their ambitions escalated. New facilities appeared, classified campuses in Patagonia, under the Siberian permafrost, and most recently, on a small island in the Andaman Sea, off the record. The name of the facility was erased from official records, its existence buried under layers of environmental research exemptions and maritime zoning laws. Satellite imagery was scrubbed. Naval patrol routes were quietly altered. On paper, it was nothing more than an ecological observatory, a station for monitoring coral bleaching and sea level rise. In reality, it was something else entirely. Something audacious, something dangerous. Because what began with rodents and rabbits did not end there. Genova had no intention of stopping at the Holocene. They were going back further, much further, deep into the Mesozoic. Dinosaurs. The word was never used in official documents, too sensational, too evocative, but everyone knew. Inside Genova's secure networks, euphemisms proliferated: Archosaurian genome recovery, deep-time biodiversity initiative, Mesozoic reconstruction. Grant proposals spoke of modeling macroevolutionary processes and restoring ecological resilience. But behind the jargon, the truth was stark. Genova wasn't bringing back recently extinct species. They were resurrecting the vanished giants of a world lost for over 65 million years. The process began not in a jungle, but in a sterile white laboratory lined with negative pressure airlocks and banks of cryogenic freezers. The key was purity. Previous attempts at de-extinction, the kind paraded by biotech startups for headlines and venture capital, were crude approximations, genomic patchworks assembled from living relatives, a mammoth spliced with an elephant, a pigeon with CRISPR-modified genes. But Reinhardt was obsessed with the original blueprint. He wanted the real thing. His breakthrough came from an unexpected vector, apatite nanocrystals in fossilized bone. While DNA degrades within a few thousand years under normal conditions, certain mineralized structures, tiny lattices of calcium phosphate, act as microvaults, preserving molecular fragments at the nanometer scale. Reinhardt's team combined laser desorption mass spectrometry with quantum coherence sequencing, a bleeding-edge technique that reconstructed entire genomes from billions of shattered fragments. Where others saw degradation, Reinhardt saw a jigsaw puzzle, one solvable with enough computing power and machine learning. The problem wasn't just DNA, it was the epigenetic code, the chemical modifications that regulated gene expression. Without it, you could clone a body, but not behavior, not instinct, not life as it once existed. Genova's answer was audacious. They mined molecular imprints from fossilized soft tissue, then inferred the methylation patterns through comparative analysis with extant archosaurs, birds, and crocodilians. The result was a functional genome as close to the late Cretaceous original as modern science could achieve. In 2016, the first embryo was grown inside a custom bioreactor, an artificial womb that simulated the gas exchange and pressure fluctuations of a Mesozoic egg. It survived 38 hours before catastrophic organ failure. 100 more failures followed. Then on attempt number 142, a creature hatched. It was no bigger than a chicken, but it was real. That moment triggered an avalanche. On Ko Sangraam, far from prying eyes, Genova constructed an ecosystem as bold as it was reckless. Sprawling enclosures that mimicked ancient floodplains, mist-drenched aviaries that roared with wing beats, subterranean aquariums where armored leviathans drifted in darkness, greenhouses cultivated ferns, cycads, and horsetails, flora resurrected from carbonized spores. Everything, plants, insects, microorganisms, was cloned or genetically reconstructed because Reinhardt understood the brutal truth. You cannot recreate an apex predator without recreating its world. For a time, it worked. Behind walls of armored glass and redundant security protocols, the impossible had become reality. Investors, billionaires, pharmaceutical CEOs, military procurement officers walked the observation corridors in hushed disbelief. They peered through reinforced panes at shapes moving in the mist, scaled silhouettes, feathered shadows, the slow ripple of muscles sheathed in ancient architecture. Creatures unseen for 66 million years now walked again, their existence a defiance of extinction, a negation of time. The project bore the codename ARC, Adaptive Reinstatement of Clades, a sterile acronym designed to pacify regulators and lull compliance officers. On paper, it was a biodiversity restoration initiative, a contribution to the genomic resilience of Earth's ecosystems. In truth, it was nothing less than the most audacious experiment in the history of life sciences, a reassembly of genomes, atom by atom, base pair by base pair, until deep time itself yielded. Internally, ARC was marketed as a miracle engine. To pharmaceutical conglomerates, it promised compounds beyond the reach of contemporary evolution, peptides and enzymes shaped by ecosystems more competitive, more brutal than anything known today. Proteins hardened against extremes of temperature, salinity, and pH. Cell membranes resistant to viral infiltration, novel metabolic pathways capable of synthesizing biopolymers unknown to modern chemistry. To the energy sector, Reinhardt dangled an even greater lure: alternate biochemical energy systems. Dinosaurs had thrived under atmospheric oxygen levels far higher than today's 21%. Their respiratory adaptations, their hemoglobin efficiency, hinted at a biochemistry optimized for performance in low oxygen, high CO2 environments, the exact conditions projected for Earth's future climate models. Nature solved problems 60 million years ago that we haven't even begun to ask, Reinhardt said in one presentation. The answers are written in their DNA. To military contractors, the appeal was more primal: shock organisms, bioweapon countermeasures, armored integuments. The language was careful, couched in euphemisms, but the meaning was clear. These animals were test beds for technologies no defense lab could synthesize: adaptive camouflage, natural ballistic shielding, tissue regeneration beyond mammalian capability. And always, Reinhardt stood at the center of it, the consummate showman in a white coat, his voice a measured cadence of authority and seduction. He spoke not of monsters, but of molecular miracles. He invoked the poetry of evolution. 3 billion years of iterative design, perfected by extinction bottlenecks, refined in ecosystems harsher than anything our planet now sustains. His favorite line, repeated with theatrical gravity: "Every organism in this facility represents a library of solutions, solutions to problems we don't even know exist yet. The next antibiotic, the next biomaterial, the next energy pathway. It's all here, written in code, older than the Himalayas." And for a time, they believed him. The investors, the auditors, even the scientists who wondered in the privacy of their thoughts if they had crossed a line too far, too fast. Beneath the sleek marketing decks and the choreographed investor tours, reality was something far uglier. The early phases of the program were a blood-soaked exercise in trial and error, a stark reminder that life, once broken, does not yield easily to reconstruction. On paper, the models were elegant, recombinants mapped across terabytes of genomic simulations, predictive phenotypic algorithms running on quantum clusters. But the wet lab was a different world, where mathematics met meat. Minor missteps, single nucleotide insertions, epigenetic silencing errors could cascade into catastrophic outcomes. The genome was not a code to be cleanly edited. It was a living labyrinth, a system honed by eons of evolutionary trial and error. Break the wrong feedback loop, and you didn't just get a dead embryo. You got something else, something wrong. They kept the failures hidden, stored in refrigerated morgues marked in stark red, biohazard. There were hundreds of them, rows of stainless steel drawers lined with polyethylene bags, each containing something that might have been alive. Some never made it past the blastula stage. Others hatched. That was worse. Malformed hatchlings emerged from synthetic eggs, slick with amniotic gel, twitching in the gloved hands of technicians. At first glance, they looked almost plausible. Tiny, reptilian, something that could pass for prehistoric. But then the details emerged. Fused vertebrae, blind sockets where eyes should be, limbs conjoined in grotesque configurations, jaws frozen in rictus, needle teeth exposed in permanent agony. And still, some lived. They lived long enough to move, long enough to scream. The shrieks were primitive, high-pitched, and piercing, like a modem wailing through flesh. Monitors spiked in response, heart rate erratic, oxygen saturation plummeting as internal organs collapsed under the burden of incompatible blueprints. Their circulatory systems couldn't sustain the metabolic load, livers ruptured, lungs failed, tiny hearts tore themselves apart trying to maintain coherence in bodies that were never meant to exist. The technicians documented everything: pulse, respiration, blood chemistry, because Reinhardt insisted on it. Every failure is data, he reminded them, voice clipped, eyes cold behind wireframe glasses. Every anomaly teaches us something about the architecture of life. But in the quiet hours, when the lights dimmed and the filtration systems hummed like distant surf, some whispered that the anomalies were teaching something else, something they didn't want to learn. Because patterns were emerging, not random, not noise. Patterns in bone morphology, in neurogenesis, in the way certain species exhibited reflex arcs no one had programmed, things adaptive, things exploratory. And that was the part no one talked about. Not in reports, not even in whispers. The truth was undeniable. The genetic code of deep time was not a tidy archive. It was a corrupted lattice of broken sequences, riddled with noise and voids. 66 million years had not been kind. Genova's molecular biologists learned the hard way that DNA was not a set of instructions. It was a dynamic, self-regulating system. And when you grafted fragments onto living scaffolds, the system fought back, hard. Every step forward was a negotiation with entropy. Polymerase enzymes stalled mid-replication. Epigenetic markers misfired. Critical regulatory genes, Hox clusters, homeobox sequences, behaved like rogue software, rewriting skeletal plans without warning. The immune systems of their surrogate hosts collapsed under the biochemical onslaught. Reinhardt's team, facing escalating investor pressure and the looming threat of bankruptcy, made the decision that would damn them all: synthetic bridging. Instead of restoring genomes with perfect fidelity, a fantasy, they began to splice in fragments from extant species. Avian DNA provided missing regulatory sequences, reptilian fragments patched structural gaps. At first, it worked. Embryonic lethality rates dropped. Hatchlings survived. But they were not authentic, not really. They were variants. At first, the variants seemed stable, superficially so, until behavioral anomalies emerged. Extreme aggression, far beyond any predictive model, hypermetabolic states that drove relentless feeding, neurological instability manifesting as cannibalistic frenzies, stress response pathways so exaggerated that tranquilizers failed entirely. Worse, some displayed traits no algorithm foresaw. Accelerated ontogeny, adaptive pigmentation, novel dentition. They learned fast. They probed containment systems with almost mechanical persistence, exploiting microfractures in polycarbonate panels, testing fence voltages during power fluctuations. Hubris is a virus, and on Ko Sangraam, it replicated unchecked. The incident, later sanitized in Genova's official dossier as a containment anomaly, was anything but minor. Classified telemetry fragments tell the story. A Category 3 tropical cyclone hammering the Andaman Sea. Wind shear exceeding design tolerances. Backup generators drowning in storm surge. One by one, containment fences flickered and died. Within hours, the island plunged into a blackout. Then the telemetry stopped altogether. Personnel logs show 38 names. None returned. Thermal satellites recorded blooms of heat signatures moving inland as structures ignited. 3 days later, smoke plumes coiled skyward, visible for 50 nautical miles. By the time Genova ordered a full scrub, their euphemism for asset liquidation, the island was already lost. They buried it all, redacted reports, financial ledgers, satellite imagery. The coordinates of Ko Sangraam were quietly purged from shipping maps, its approach restricted by marine conservation statutes. But extinction is a one-way door until it isn't. The variants, forged in heat and scarcity, endured. Some bred, others hunted. Over time, the ecosystem warped into something unrecognizable, a self-reinforcing feedback loop of predation, mutation, and selection pressure. What emerged was not prehistory reborn. It was something else entirely. A new biosphere, weaponized by time and error. An apex predator's paradise beyond human dominion. And it was still evolving. By 2022, the name Genova was little more than a whispered cautionary tale, a corporate ghost buried under layers of litigation and non-disclosure agreements. The headlines had vanished, the lawsuits sealed, the leaked photographs scrubbed from every darknet forum. To the public, Genova never existed. To the scientific community, it was an urban legend. But the story didn't end there. Genova's intellectual property, patents, sequencing algorithms, and terabytes of genomic reconstructions didn't disappear. They were quietly purchased in a series of opaque shell company transactions, routed through offshore accounts and cybersecurity proxies. The buyer? NeoGenix Industries, a privately held conglomerate with headquarters in Luxembourg and research campuses scattered from Singapore to São Paulo. NeoGenix was not just another biotech firm. Its founder and controlling shareholder, Cyrus Phillips, was a billionaire visionary with a taste for risk and a disdain for regulation. Where Genova had been secretive, NeoGenix was invisible. Where Genova had hidden behind layers of scientific pretext, biodiversity restoration, pharmaceutical innovation, NeoGenix operated without pretense. Inside the boardrooms, the narrative was simple. Genova didn't fail because the science was flawed. Genova failed because its leadership panicked. Poor containment, inadequate redundancies, catastrophic decision-making. These were managerial problems, not technological ones. Problems NeoGenix believed it could solve. So they started again. Under a new name, with deeper pockets, hardened security, and a team of geneticists drawn from the bleeding edge of synthetic biology, they secured next-generation quantum sequencing arrays, CRISPR-CX editors capable of altering entire chromosomes in a single pass, and AI-driven protein-folding simulations to map metabolic pathways that hadn't existed for 66 million years. They weren't just reviving Genova's dream. They were perfecting it. There were no press releases, no environmental impact statements. Every server was air-gapped. Every shipment moved under diplomatic exemption. And deep within the Congo Basin, on a parcel of land scrubbed from every navigational chart and listed in no registry, construction crews broke ground on something that would make Genova look like a proof of concept. This time, there would be no warning. This time, the world wouldn't even know until it was far too late. In the end, the story of genetic engineering is not just a story about science. It is a story about power, about humanity's insatiable desire to control nature, to rewrite the rules of life itself. We are standing on the edge of a precipice, and the decisions we make today will shape the world of tomorrow. But as we forge ahead into this new frontier, one irrevocable question remains. How far are we willing to go? And when we cross that final line, will we be prepared for the consequences?